Protein purification in a little over a minute. The protein purification process begins with an Avigel blue column, a matrix of sepharose beads that has a special dye in it that binds to the protein and gives it its deep blue coloration. The sample that contains the protein to be purified is added to the top of the column and allowed to bind to the dye. The sample passes through the column and the rest of the liquid in the sample is drained out, since the protein we want is bound to the blue in the column. A solution with a different concentration of salt is applied to the column to wash out more of the undesired protein. Another solution with a high concentration of salt is applied to the column. This causes the protein we want to unbind from the dye and flow out. While this is happening, a fraction collector drops the outflow into glass tubes and shifts to the next one once a certain volume has been reached inside a tube. The protein content of each collected tube is determined and the tubes at the peak are mixed together. The process is then repeated with a single-stranded DNA cellulose column, which has the protein bind to DNA instead of dye. This process follows the same steps as the Avigel blue, with sample application, a wash, and then collection of more fractions. Protein content of the fractions is determined once again, and the tube at the peak moves on to the final step. The last step involves a Q-Sepharose column that's much smaller than the other two. This column requires many additions of buffers, with slightly increasing salt concentrations to get the protein to flow through. Since this is so small, I can't use the machine and need to manually switch from tube to tube. Once all of the fractions have been collected from the last column, I freeze them in liquid nitrogen to make sure the purified protein doesn't degrade. And that's it. Thanks.